Chapter 1, The Microbial World. This chapter is an introduction to microbiology and kind of highlights a lot of the topics that we will be discussing over this course. So I'm showing you a few interesting facts here just to let you know that not all bacteria and not all microbes are bad. They kind of get a bad rap, but they're actually very important when it comes to the environment, when it comes to life cycles of all organisms, and they really do help us out a lot. Another interesting thing about something like bacteria and other organisms is that some can live in very extreme environments. And so most of the ones that we study probably survive around room temperature or body temperature, but there are some that have adapted to live at high temperatures, extremely high temperatures, and extremely low temperatures too. Also, they can move. Sometimes they move because we pass them on, maybe through a sneeze or a cough. But also there are some that actually have little flagella, little things on the outside of their cell that allow them to actually move and to swim. So we'll be learning about that as we go. So when we study biology, we're studying living things and how they interact. But when we study microbiology, we're studying these things, but really those things that are very small, generally that we cannot see without using a microscope. So when it comes to microbes in our lives, some of the different ones that we'll be studying, again, these are things that are too small to be seen without something like a microscope. You probably heard of bacteria. When we study protozoans, we'll study little single-celled organisms. Maybe you've seen an amoeba or a paramecium. Algae, there can be single-celled algae, little organisms that swim in the water, or there can be multicellular algae, which is not what we study as much in this class, but something like if you're swimming in a lake and it's wrapping around your legs. When it comes to fungi, we'll be looking at things like mushrooms and molds, but also yeast falls into that category. Viruses you probably have heard of. They're kind of a special case that we'll study. And then helminths, you may not have heard this term before, but you probably have heard of something like a tapeworm or a roundworm. And while these organisms are not small themselves, their eggs are very small, or their larva is very small, and it's easy to pass those items from one organism to another. So like I said, usually microbes are often associated with have a negative connotation to them. If they are capable of causing disease, we call these pathogens. So we might say this bacteria is pathogenic, but there are a lot out there that are not. So as I mentioned briefly, there are some that really are important to us in the environment. They break down substances that other things cannot break down. They can be used in bioremediation. For instance, when we clean up oil spills, a lot of times there are certain bacteria that would prefer kind of an oily fuel to eat, and they prefer to break it down. So if you put those into where there is an oil spill, they can naturally kind of clean it up. Photosynthesis. Photosynthesis is using light and carbon dioxide to produce energy and oxygen. So when it comes to something like some of these little algae that we'll study, there are a bunch of algae living on the surface of all of the water in all of the earth. And even though they're tiny themselves, they actually produce more oxygen than all plants that are living on land. So while we don't see them very easily, they're definitely very important and kind of cool. So what we call living on us is our normal flora. So like it or not, you are covered with bacteria, fungi, protozoans, all these different things. And again, most of them do not cause disease. But what they do is if something pathogenic comes along, they're going to try to compete with that microbe. They don't want it to take over their space because they're having a nice environment to live, to find food, to reproduce, and they're living a nice life. So they do protect us from disease, and we'll talk about that later as we go as well. So definitely they help us in our respiratory tract, our digestive tract, and then they're living on our skin. But basically you're covered. There are also commercial uses. So any organism, any cell makes products, and sometimes these products are used to actually make things that we want. So when it comes to a fermentation process, you probably have heard of yeast, which is a type of fungus being used to make bread, or to make alcohol, to make beer, to make wine. And so also there are other things, preservatives that these microbes make to help us keep our food longer, and different products like vitamins and whatnot. So be sure you read more about those. So when it comes to science, we need to talk a little bit about science and we'll talk about what is good science as well. You probably hear a lot of things and it's hard to know what to believe. But in general, science is when we're trying to figure out how things work around us, and it's based on questions. And so there's kind of two major approaches. One is called discovery science, where we see new things and we learn about new things, and it may just be by watching or observing. But there's also hypothesis-driven science. And so a hypothesis is an explanation that we think might make sense when we ask a question, and that hypothesis has to be tested. It has to be tested, it has to be supported, there has to be abundant evidence that supports that if it's going to become a theory. If it becomes a theory, then that's usually the accepted explanation of the question that we've asked in the first place. It doesn't mean that it might not be disproved later, may not be supported later, but it is backed up by good scientific data. So when scientists are studying, then they use something called the scientific method. There's a lot of ways to talk about the scientific method. Maybe you've had to memorize steps of the scientific method before, but basically there's kind of some themes that are talked about all throughout. And so usually there's some kind of observation. After observation, a scientist asks a question that this person wants to figure out what's going on. And so they may form a hypothesis, and they may form a few hypotheses of what they think is actually what it is or what is going to work, or what the cause of something is. And so after that, the scientist would design an experiment to test out the hypothesis. And so after an experiment is done, it has to have a lot of subject matter in it, has to have a lot of repeats, it has to be repeatable by other groups that have not, decided to do the experiment in the first place. And so if you have all of these results that add up, then you can say you support your hypothesis. Your hypothesis is supported, and the data supports that. Sometimes you need to go back and revise a hypothesis or retest, because maybe somebody has discovered that, oh shoot, I was testing, but I had too many variables, and that doesn't make any sense, and it didn't really give me the information that I needed. So that's okay to do. And so a lot of times scientists have to go back to the drawing board over and over again before they come up with anything of substance. So once someone comes up with a theory, then it has to be subject to peer review. So these experiments would have to be reviewed and talked about by other scientists to make sure that there was no bias. And then, like I said, these experiments need to be repeatable for other groups to do and get similar results. So now we'll talk about some of the scientists in history. There are quite a few, but we're not going to talk about too many of them. But the ones we do mostly have their contributions related to microbiology. So the first scientist is Robert Hooke. And you can see from the dates here, I'm not expecting you to memorize any dates, but I want you to realize that these discoveries occurred, several hundred years ago. And you'll also see that things take a long time when it comes to discovering new things. But if you think about the 1600s, the 1700s, even the 18 and the 1900s, we don't have the advantage of modern transportation that we have today. So it takes a long time to get information from A to B. So Robert Hooke is kind of credited with learning about cells in the first place. And so he was using a microscope, and that microscope was allowing him to see things on a larger scale. And he was studying cork cells, which is a cork is a type of plant. And he saw that there were these divisions. And so you can kind of see in this little picture here, just these little spaces or compartments. And he eventually called them cells. And so he had to kind of study the world around him. He studied things that were growing around him. It's not like today where you can just call Carolina Biological Supply and order something up to study. And so he had to use what he had, and that's what he did. There's also Antony von Leeuwenhoek, and notice this is still the 1600s to 1700s. And he built his own microscope, and you can kind of see it here in figure B. It doesn't really look like a whole lot, but it did allow him to magnify things and see them very well, much better than he could as well. Again, he also didn't have a lot to work with, and so he studied cells around him, things that he had. Insects, things that are in pond water. I've always heard he studied his own feces. He studied sperm cells. He saw under his microscopes that there were things that were moving. And so back then, living things were either plants or animals. And so he called them animalcules, just to indicate their movement. Again, we're talking about a long time ago, and so this was a big discovery for that. I'm actually reading a book that tells a lot more about him and how he made all kinds of microscopes so that he could leave his specimens underneath them because he didn't want to take them down. He wanted to look at them all the time. Okay, then there's Louis Pasteur. You may have heard of pasteurization, which he is credited with as well. Notice we've made a jump here to the 1800s, almost 1900. And so he's credited with pasteurization, like I said. And basically what that is that's taking a substance like you probably heard of it with milk or juice, but it's giving it a heat shock. And so there's naturally occurring microorganisms in the liquids. And so because of that, they will spoil pretty quickly. So as far as trade goes, they were trying to ship things from across the ocean, and things were spoiling before they even got there. And so he tried this procedure where you just give it a little bit of heat. It doesn't kill everything, but it kills a lot more of the microorganisms. And so they don't grow as fast and spoil. If your milk was not pasteurized, it wouldn't last very long. But most of the milk we buy is pasteurized, so it lasts a lot longer for us. He also had this idea that microbes cause disease. And so he developed what is now called the germ theory, basically that a certain germ causes a certain disease. And there was a thought out there called spontaneous generation. A long time ago, people didn't really have a lot to think about science. They wanted to find explanations for things that happened, and so there was this theory called spontaneous generation, that living organisms just kind of appeared magically. And so it took a long time and a lot of experiments between a lot of scientists to actually disprove spontaneous generation. So eventually the theory of biogenesis held to be true. And so Louis Pasteur was involved in this along with other scientists and with his famous S-shaped flask. Make sure you look at this in more detail in your textbook, but basically this experiment goes through where he had a sterile broth, it was open to the air, and over time microorganisms appeared in it. And so when he actually sealed it off, then there were no microorganisms. So the top picture shows it open, there were microorganisms after time. The second one was sealed, and there's no microorganisms. And so a lot of times people would just say, well, it magically came from the air and got in there. Well, so then he had to use this S-shaped flask, and that actually, the air was not able to get in there, and so there were no microorganisms over time. And eventually, once if you cut the neck and you allowed the air to get in there, then the microorganisms could come as about. But as far as spontaneous generation, there was a definite reason why they actually appeared, and it wasn't just because. Robert Koch, or Koch, 1800s to the early 1900s, like I mentioned before, the germ theory of disease. He looked at looking at specific diseases and he was able to find a set of postulates that could prove which microorganism caused which disease. And so you may have heard of anthrax before. It's actually comes from a bacteria, Bacillus anthracis. They form these endospores, which are hard, desiccant, dry capsules, and they can be passed on from one source to another. They can last a long time. They can last up to thousands of years. They don't really die because they're in such a dried out state. But if we were to inhale them, then they would rehydrate in our body and then cause the disease anthrax. And so he studied this disease in particular. He came up with a set of postulates, or kind of a test, to prove that certain microorganisms caused a certain disease. So if you look at figure 1.8, this kind of shows you his experiment. This is not possible to do with everything, but sometimes it is that simple. In this case, you have a diseased animal in picture 1 there, and so you take the microorganisms from that diseased animal, you look at them, you inject them, you isolate them, you inject them into a healthy animal, and you see if you get the same disease. If you get the same disease after looking at it again through the microscope, then you can say that that certain microorganism caused that disease. Make sure you look into this a little further as well.